your hand, and that's, believe it or not, a microscope. And the first microscope he built that allowed him to take a little bit of his saliva, put it on that little dish at the end of this butterfly-shaped rod, that is what you focus with. And what he saw were these little creatures. Can you see my pointer?
Yes.
Okay. What he saw were these little creatures that he called animalcules, small animals. And the reason he understood these were creatures as opposed to debris or not non-living things was because they were frantically moving anytime he tried to look at them. And that's reportedly the first observation of microbes, and specifically of, in this case, most likely bacteria. Now, microbes and immunology, I'm an immunologist by training, and even more than by training, by mentality probably. And the relationship between immunity and microbes has always been a special one, and a highly intertwined one from the very beginning. I call it a special relationship because, in fact, early on, it was even difficult to decide whether a given scientist was an immunologist or a microbiologist, which is, I think, telling in and of itself. And if you look at recent reviews or reflections that have been made over this, you see this theme coming up over and over again. So Paul Ehrlich, translational medicine, depending on where you look, is defined as a microbiologist or an immunologist, because of course he defined the fundamentals of immunology, but also identified pathogens. Same for Pasteur. Who doesn't know Pasteur? Well, Pasteur created and formulated the germ theory of disease, but he's also, of course, an immunologist, and that's how, unless we have appropriated him, but I think he was thinking about himself as an immunologist. Not to mention Mechnikov, who is more and more on everybody's mind right now, because he is the first one who really formulated the notion that we, not only we, but we as well, are endowed with a kind of natural immunity, as he called it, which we now call innate immunity, the main purpose of which, of course, is to detect microbes, not just pathogens, but microbes. Now, this relationship has, however, over time, taken a specific flavor, which is that microbes, animalcules from Leeuwenhoek, are something that we have to defend ourselves from. And this sort of culminated in it, you could say, when we as a community, and particularly Alexander Fleming, who you see bowing in front of King, I think Gustavus Adolphus of Sweden in 1945, getting his Nobel Prize. This is Einstein in the back, for his discovery of penicillin, namely an antibiotic that could, in fact, get rid of microbes. So, and the interest was enormous. And we are talking at the time when a war had been raging, and as you know, and that has been assessed very rigorously, more people died of their wounds and of the infection of their wounds that they actually died directly on the battlefield. So this was an enormous problem. And when the medicine started understanding better how microbes lived, where they lived, how they were passed from one person to the other, of course, the number of infections started decreasing, mortality started decreasing. I mean, there is a very vast literature showing, in essence, that microbes, specifically bacteria, but not just bacteria, are our foes. The question is that always invariably and clearly true? or is this a little more complicated than that? And obviously, I wouldn't be here talking about this if this weren't the case. And so I'm going to tell you my own story, my own scientific story, which I think captures the essence of the special relationship between microbes and immunity in a way that proves that, in fact, microbes are indeed something that is very, very good to have around ourselves, at least some of them at some time. And this story, oh, by the way, you can stop me anytime. I won't lose my train of thought, hopefully. So if there is something I say that isn't clear, you don't have to wait until the end to ask. Sorry if I didn't say this earlier. So I'm going to tell you a story that has to do with the type of complex disease. As you heard, I am the director, and I am especially indebted to Leslie Tolbert for that, because it was under her inspired leadership that happened. Of this somewhat virtual, but not completely virtual center that is interested in complex disease biology. And asthma, we always quip, is the most complex of complex diseases because it is really difficult to understand. But at the same time, is also extremely important to understand, if only because it is the most common chronic disease of childhood. Now, if you look at the distribution, epidemiologically of asthma, childhood asthma in the world, and you immediately see the coding on the left in terms of percent of the population. This is, in fact, not just children and adults together. And what you see is a lot of red wherever people speak English. So much so that a friend of mine, who is arguably perhaps one of the greatest asthma geneticists, Bill Cookson, always says that asthma is a disease of English-speaking people. But he says that as a joke, but it is true that countries where English is spoken either as a first language or as a main language are typically very high in their prevalence of asthma. Now, the point, of course, is nothing to do with English per se, and a lot to do with the lifestyle that accompanies this feature of which language you speak. And in other words, it's a way to say that countries that are very westernized in their lifestyles are also the countries in which the prevalence of asthma is the highest. And this concept that asthma is an environmental, to a large extent, an environmental disease, has been first brought home in the early 2000s, particularly by this paper, which I always like to show, by Jean-François Bach, who, of all places, was at the Institut Pasteur, so a very becoming source, given what I said initially about the relationship with microbes, between microbes and immunologists, and showing this very rapid increase in asthma prevalence over a relatively short period of time in all the records that he could get his hands on. You see this tripling, quadrupling of asthma prevalence over a period that is more or less 20, 30 years. Now, despite the fact that many of us, we included, are extremely interested in asthma genetics, genes by themselves, their mutations by themselves, or variants by themselves, can by no means explain a rise that rapid in anything biological, and certainly not in a disease phenotype. So this is a visually powerful, but also conceptually very compelling pointer to the fact that asthma is an environmental disease. Now, we all know more or less scientifically or intuitively that there are a lot of things that are bad for asthma in the environment. Certainly, if you are an asthmatic and you live in a highly polluted environment, that doesn't do you any good. If you inhale a lot of dust during a dust storm or things like that, not to talk about pollens and all that. But the story that I'm going to tell you today is the flip side of this coin, and this asthma and the environment in their opposite orientation, so to speak. In other words, the way in which the environment can teach us about protection. And this is a story that starts in the regions of Europe approximately 15 years ago, where a group of very smart, curious epidemiologists observed something very counterintuitive at the time, namely that the prevalence of childhood asthma among children who were raised on very traditional farms, on the kind that you encounter if you go to Bavaria or you go to Austria, particularly in Tyrol or certain regions of France, Italy is the same, Switzerland, was extremely, extremely low. And they grabbed this observation, didn't know what to do with it for quite some time because it wasn't clear what it meant. And so being very good scientists and very precise in their approach to science, started dissecting what type of exposures might have explained this extremely low prevalence of childhood asthma and all the associated diseases and phenotypes, so allergies and hay fever and all that in those children, and started honing on a few characteristics of those environments that were particularly important, which you see recapitulated in this picture that one of those very good epidemiologists and one of my closest friends, Erika von Mutius, captured in this very, very clearly. So you need to be a small child, first of all, very, very young, the very first year or years of life. That's when these exposures have to be there for you. And these exposures are, well, cows. So it so happens that these traditional farmers in whom we see this protection are farmers in dairy farms. So these are cows that are there to produce this stuff, namely this milk that these children consume, sometimes even unpasteurized, because they trust it. They know their cows, they know that these cows are healthy, so they just keep the milk in the fridge and give it to the children. And you have to be exposed to the hay that these cows use to sit on and to do other things on that you see in the back. There is a lot of dung here. You can smell, by the way, these villages before you see them when you visit them. And the other very important thing is that this exposure has to occur not only in the child, but probably in the mother. This is the mother of this child working in the back here. And in this very traditional style of life, these mothers who work on these single family, small farms, work during pregnancy. They certainly don't stop during pregnancy, and they therefore are exposed to the same environment as their children, and their developing child, the child in utero, is also exposed to the same environment. So all of this converges to give you this extremely low prevalence of asthma. That was the idea epidemiologically. Now, the problem with these studies, the studies were enormously influential. And by the way, they led to the formulation, to the identification of what is called as the farm effect. Farm effect means being protected from allergies, from asthma, if you grow up on a traditional farm. Now, the question was, how do you really get there? What is really at the root of the farm effect? There were a lot of questions that remain to be answered. First of all, there was a question about genetics. Are these, because these are all relatively isolated populations, and so how applicable is this to populations living somewhere else? After all, we're talking about the alpine regions of Europe, so is this a relatively limited phenomenon? And the answer to that was no, and is no. Eric and I wrote a big review for Nature Reviews Immunology that has at this point, I don't know, 800 quotations, because we went through all the studies that had been published at the time. And essentially in every single one, whenever the exposure was not just to any farm, and we will get back to this concept, but to a traditional farm, small farm with lots of cows, where the children roam the land freely from very early infancy, the protection was there. But there were questions about genetics that were unanswered. There were questions about mechanisms that were unanswered. And so this was always a very interesting topic that, however, in a sense, didn't fully mature, I would say. Which is why at some point we sort of decided to take things into our hands. Because again, as an immunologist, I always thought that there was something hidden in this farm effect, which was fundamentally important and that deserved to be teased out. And so we, through a series of fortunate coincidences, which I will tell you about if we have time later, if you want to know, but I will sort of gloss over right now because otherwise it would take too long. We identified a model that could take care, in a sense, of many of the questions and could answer, not take care of, but answer many of the questions that were lingering about the farm effect. And this model was offered to us by a direct comparison of Amish and Hutterites. Now, I'm sure you know who these people are. Let me give you a sort of 101 course about them. Amish and Hutterites are German in terms of their origin, Anabaptists, who, because of religious issues, were persecuted very, very relentlessly and pitilessly in Europe in the 1500s. Very close in their habits, in their beliefs. As I said, they're Anabaptists, and very even close geographically. The Amish originated from a group in the area around Zurich, Switzerland, whereas the Hutterites originated from a group around Linz in Tyrol. And as the crow flies, as a bird flies, we are talking about regions that are separated by 200 miles. So little more about the distance between Flagstaff and Tucson. This is how close these people were in the heart of Europe. They ran away, they migrated around Europe, no one wanted them, nowhere they were safe, so finally they got on a ship, they came to America, and they finally settled in different parts of the North American continent in several places, and the ones we had a chance to study were the Amish from Indiana and the Hutterites from South Dakota. Now, Amish and Hutterites are a perfect population for us to study, we think, because they share so many characteristics that are critical to the argument that we were trying to illuminate. First of all, these are both populations that live on farming. We'll get back to that, but generally speaking, they would define themselves as farmers. They have very similar family sizes. This is important in asthma epidemiology. We know that a very large family size is protective, and the number of children among the Amish and the Hutterites is very high, 8 to 10 per family, and comparable. They share their diets, a very heavy Germanic diet, high calorie. They don't let pets in their homes. They don't give, unfortunately, a very extensive education to their children. They send them to school until age 8 and until 8th grade, and then it's off to the fields. They don't like internet. They don't like much of media, even though they, of course, they read newspapers, but they are not very much, certainly they don't spend hours in front of the screen as we do these days, especially. And they even speak dialects that are very similar, even though they have diverged in terms of their life for so long. And they're all, of course, based on an ancient version of 1600 version of Germanic Southern dialects. We could ascertain that they are comparable genetically, which was important for us to really sort of determine. And if you look at these in this sort of space of different genetic populations at these different, this is a principal component analysis, suffice it to say, every person is a dot, and every color is a person from a different part of Europe. And you see that the Amish, which are going to be always blue in this presentation, and the Hutterites, who are going to be always red, are smack on top of one another right here in the middle of this graph. And they are much closer to one another than they are to any of the other populations in this graph. And so some of them are Tuscan, I'm from where I come from, I'm a Florentine. Some of them are from the Basque Country, some of them are from France, some of them are Russian. They are all very highly dispersed in this space, whereas the Amish and the Hutterite are very similar to one another again, and different from the other population that were looked at. But, and I'm sure you were expecting my but, they are different in one way, which is critical and is why one population is, we think, a great, if not perfect, control for the other. I said they are farmers, and that's absolutely true. However, very different kinds of farmers. The Amish here on the left are your traditional farming communities, as I described them to you. They farm using horses. They have no equipment. They have dairy farms that are basically family farms. And when we went and visited them, Erika von Mutsus and I, Erika told me this is like going 250 years back in time. These are the old European dairy farms of the early 1800s. That was her comment. The Hutterites, on the other hand, are extremely entrepreneurial farmers. They also are farmers. They are also dairy farmers. But instead of having your single family farm with 15, 20 cows, they have these very large hangars where they have sometimes hundreds, sometimes even thousands of cows. And of course, what that implies is that the way these cows are handled is very, very different. And what that implies even more, perhaps, is that the way the children, because we are talking children here, who grow up on those farms, and the type of contact that they can have with these animals is very different. So this is, note, this is the same scale in these two photos. And this is your typical Amish farm. It might be the one we go to and spend time in whenever we visit these populations. And here is your house. This is the barn. This is the stable. You cross the courtyard, which is usually unpaved. This is a sort of, a dirt road. And the child runs back and forth as they do. They spend most of their time playing when they're small children in the barns, in the stables. The cows are not off limits, in fact. And they play with them, or they help their father milk them when they're able to. In other words, there is a very large level of exposure for these children to those cows. This is a communal Hutterite farm, and the farm among the Hutterites are, as I said, they're called colonies. And this is what is striking. So these are homes, and these are townhomes, one close to the other, a bit like in England, some places. Every family has a home, obviously. They all eat and pray and do things together in this building at the center of the colony. They have shops where they fix their equipment, where they keep their, machinery and so on, like that. In fact, the Hutterites have developed a lot of machinery, heavy equipment, because if you have very large farms like that, you need that. They drive. They certainly don't use buggies. They don't use horses to farm. They use machinery like in the more industrial farm, and that's the key word. But if you ask me, where is the barn and where are the stables, on this photo, I would have to put my pointer out of my screen. In other words, they are far away. And they are far away in part because they are so large, in part because they are perceived as being places where there is some danger, especially for the kids to get in. And so basically kids won't get into those barns and stables until they are 10, 12, 15 years old. And that happens to be, frankly, too late for those exposures to have any effect. Now, you could say, well, all right, does it really matter? Well, it does. It does because we could sample the environment, and particularly the microbial environment among the Amish and the Hutterites in their homes. And you know, homes are your home, but it's also where you bring things from the outside. It's amazing how many bugs we find in those homes, in particular among the Amish, that actually come from the barns and from the stables, because they come in with your on your clothes, on your shoes, and so they actually, you import them. And sure enough, if we compare, this is endotoxin. Endotoxin is a microbial component that we use as a proxy to get a sense for the microbial load in these homes. What we found was the Amish homes were approximately 7-fold higher in microbial load than the Hutterite homes. And not only they were higher, but they were very different this was a very rough first look at what is in that dust in those homes in terms of microbes. And you can immediately see, even just by eyeballing this figure, that in fact the microbial composition in terms of microbial bacterial families is very different in these two places. So there is a difference in quality, there is a difference in quantity in the microbial environment in Amish and Hutterites. Then the question is, does it really matter? Because you know, if this didn't translate into a difference in the prevalence of the diseases and phenotypes that are of interest to us, all of this would be perhaps interesting sociologically and epidemiologically, but have no bearing on anything. But that's not the case. Look at the prevalence of asthma among the Amish, and this is a very generous 5%. We were very inclusive in how we established our criteria. These criteria, of course, are the classical ones, but we, in a sense, almost stretched our imagination. We certainly didn't give them any discount, so to speak. And look at the prevalence of asthma in the Hutterites, 21%. This is 4-fold higher than in the Amish. And look at the prevalence of allergy, which is almost 5 times higher. So the Hutterites are essentially like your typical average inner city population in the United States in terms of prevalence of asthma and allergies, and the Amish are incredibly well protected from all of this. And now, if I put my MD hat now on, not only they're protected in terms of yes, no, having these diseases, but also the severity whenever they develop any such thing that might remotely resemble asthma or allergy. It is so mild that when we asked them and we interviewed them, we had to explain what we meant at times, because they had such mild symptoms that they sometimes don't even notice. Now, none of this, however, was a formal demonstration that all of this was environmental in origin. We are scientists, we are researchers, we have a lot of imagination, so we could sit down in a circle at night and come up with many reasons why this might have been the case, without the environment, and particularly microbes in the environment, being responsible. So we wanted to do the sort of knockdown experiment, you know, to really clinch it. And this is if I wanted one experiment from my whole career, entire career, to be remembered, this is it, because this is the experiment that we did, and that we did despite the fact that everybody in our group thought we would fail. And we did it, and it was extraordinary. We did something very simple. We collected dust from the homes of these Hutterite and Amish children, and particularly from their bedrooms, which is where these children, when they're very young, spend a lot of time. We took this dust, we extracted it in the simplest possible fashion, just with water, just to make it, to get to something that we could administer to, and here comes the daring part of the experiment, to mice. And we wanted to use mice because, of course, in mice, you can become mechanistic, you can get controls, you can ask questions in ways that you cannot do, and you can get causal in ways that you cannot do in humans. And so we made these mice allergic and asthmatic using the tools and tricks that we immunologists use to make these mice asthmatic. I should do asthmatic this way because it's not exactly asthma, but very close. And we simply had a group that we left untreated, or we treated with just control, and the group that we exposed through the airways, that's important, to Amish dust, and a group that we exposed to Hutterite dust. And we asked, does it make a difference in terms of how bad asthma is in these mice, depending on what we have given to these animals? Now, I wouldn't be telling you the story if the difference weren't there, but it went beyond our wildest hopes. So we are measuring the resistance in the airways, how strongly these and effectively these airways constrict when you make them constrict. And if you are asthmatic, they will constrict more because, of course, there is underlying inflammation. So you give a bronchoconstrictor to a mouse that has received no treatment whatsoever, and the response is very, very mild, almost undetectable. You give it to a mouse that you have treated with allergens quite, how can I say, energetically, and you see a lot of constriction. The more constrictor you give, the higher the resistance goes, because again, there is this underlying inflammation that you can measure very, very accurately and quantitate. You give this same bronchoconstrictor to an Amish treated mouse, a mouse that has inhaled over a period of a month these extracts of dust from Amish homes, and there is almost no response, even though you're giving the same allergen. You give it Hutterite dust, and here you go, not only you are not preventing this very strong asthma-like response, you actually exacerbate it. So the word that I would like you to remember in the title of this slide is sufficient. All we had to do to recapitulate these very complex phenotypic differences between these two populations was to give mice dust from either environment. And the giving dust from Hutterite mice made them not resistant, in fact, more prone to developing asthma and allergic inflammation. And all it took to protect these mice was to give them Amish dust. And interestingly enough, now the pointer to the microbes, these effects dependent completely on innate immunity. Remember Mechnikov, his natural immunity. Natural immunity, innate immunity, is what recognizes microbes. And if you disable that genetically, if you get mice that are missing, you don't need to look at the slide in detail, but the concept is if you disable genetically innate immune responses in some of these mice, in particular, this would be the ones on the right side of your slide. And you, here, this versus this, and this versus this. And you deprive them of the genes that allow them to respond to microbes. In essence, that's what we did. We basically deleted the genes that are responsible for the response to microbes. Now this protection is no longer there. So these animals, once they are knocked out, as we call them, for these genes, respond with no protection even to the Amish dust, the difference between this blue line here and blue line there, and the same thing in terms of what gets into their lungs. This is protection very strong in the wild type mice, and this protection is no longer there in the knockout mice. So not only the Amish dust confers protection, but it does it through pathways that are exquisitely dependent on microbial stimuli. Well, so it is the environment, and it is the microbial environment. And you know, this paper that had already had the let me brag about it just a little bit, but just to not to brag. And by the way, it's our paper, but it's a paper of a very large group of scientists, and this was really team science, because we needed epidemiologists, we needed the people on the field, we needed the contact with the communities, we needed a lot of things to make this happen. But we landed also on the front page of the New York Times, because this is the article, but it was on the front page of the New York Times. And we got on top of that, in the same newspaper, the same day, a beautiful opinion by Moses Velasquez Manoff, with whom I've stayed in correspondence. He's a very interesting guy, about this finding. And so I have asked myself, why is it that this got so much attention and so much press? Not just attention in the field. You know, if you're interested in the farm effect, okay, this is the demonstration of it. It's New England Journal of Medicine, impact factor 75, all these things. But why did it end on the first page of the New York Times? And I think the reason is that we gave a very formally tight and formally compelling demonstration of the fact that microbes can be very good for you. And so not just popular press, not just you know, the probiotic that you pick up at the shop, but really a demonstration of how important the relationship between microbes and immunity can be in these environments. So I think it's the implication that made this so eventful, so to speak, and so impactful. And you know, we're continuing these studies. They're not simple to do, but just for fun, I will tell you that we have been doggedly trying to go after what is in this dust that is responsible for these effects before anybody asks, because I know I would ask if I were in your shoes. And so we keep fractionating this dust and testing the effects of these fractions. And I can tell you that we are down to fractions now that are still capable of protecting these mice when we expose them to these allergens, even though what is in there is less than 0.2% of the total carbon content, so of the total organic matter that is in that dust. So we are really, we have got rid of essentially 99.8% of what is there, and we are still capable of protecting. So we are continuing these studies, but because of course it would be fantastic to know what it is that is doing this and how we could leverage it. But there is another question that came up, and that is in a sense intertwined with this one, and is not just about the microbes in the environment and the products of the microbes in the environment, but is the microbes that are in the host, in the child, in this case, him or herself. And it might sound like a leap of faith to you, or a sort of dissonance to you. Why are we worrying about the microbes in the child if we have because we found microbes in the environment that might be protective? But the fact is, and we just got a very large grant, in essence, to study this exactly. The fact is that what is in us, and particularly in the developing microbiome, sets of microbes in a child very early on, depends to a large extent of what is in the environment and what that child is exposed to. So these are not two waterproof systems, you know, the outside and the inside. There is a lot of communication. We certainly find human microbes in the environment, but we find particularly a lot of environmental microbes in humans, and particularly in children, and particularly at the time when the microbiome, the set of microbiota, microbes that we carry, is developing and taking shape. Look at this. These are our own data from our friends and colleagues at UCSF. These are shared taxa, microbial taxa, bacterial taxa, between the environment and stools, poop, at 1 month of age in the child. So as early as 1 month from after birth, we can detect environmental microbes in the poop of children. Okay, so I think this is important to stress, because this is the reason why you cannot not worry about the extent to which those microbes then are important in causing protection or not. Now, I also think that it so happens this is the right time to start asking these questions. We could not have asked these questions in any meaningful, well, or at least productive way, only, I would say, 10 years ago. But it so happens that we are in the midst of what we now call the microbiome revolution. And let me spend just a few minutes. I don't want to bore anybody, but I think it's important to understand why this is a question that now is tractable. And it is tractable because until, as I said, more or less 10, 15 years ago, but I would say 10 more than 15, all what we could do was, when we wanted to know what kind of microbes, particularly bacteria, there are in a given environment, what we could do was culture them. And, you know, culturing things is great, but only up to a point. And this had been known for a while. This is where, in a sense, at least conceptually, only conceptually, it all started. This is this famous paper that from 1985 that put forward this concept of the great plate count anomaly. And it's what I'm showing you. This is on the left. You take anything, saliva or any secretion from the human body or from any environment, and you culture it in a plate with some medium, and you see some colonies appear, and you're very happy about those colonies, and then you, as we all have done in our youth, many of us at least, you pick those colonies, you grow them, you sequence, you see what it is, blah, blah, blah. But if you take the trouble of looking at the same material that you put on your plate under the microscope, so doing nothing particularly elegant, you see that there is an enormous amount of this stuff around there. And so this was, you know, and we're talking orders of magnitude. And so this was what was defined as the great plate count anomaly, but people didn't know what to think about it and how to think about it, because again, you could only culture what you could culture. And so that is where you were stuck. But then something happened. And what happened was basically next generation sequencing, which is this at times insufferable, but most of the times incredibly useful approach that allows us to sequence anything under the sun, even though we know nothing about it, essentially. As long as you have a piece of DNA or RNA that you can attach a linker to, and the linker is known, now you use that linker, so to speak, that piece of DNA that you attach to the ends of this unknown piece of nucleic acid as a Trojan horse to get into what you want to know the sequence of. And so now we can sequence essentially anything under the sun, and we're doing it. And so now we have this method of detection that is culture independent, that gives you a totally complete profile of bacterial communities in a given environment or in a given sample. And you know, when I teach my complex disease course, I always tell my students that they should feel incredibly privileged to live at a time when, despite all our triumphs in science, we are back to doing, listen, here, taxonomy. In other words, every day, by doing this exercise, laboratories are reporting sequences of bacteria and other microorganisms, the existence of which we completely ignored. And they have to be put in phylogenetic trees. And you do that, of course, based on sequence. So the poor Linnaeus who had fallen into disfavor because, taxonomy was considered a thing of the past, well, now we're doing taxonomy again because we have this enormous amount of microbes that we are being discovering every single day. And by the way, to go back to the culture method, we now know that what you can culture at any given time is, on a good day, 1-2% of what is really there based on sequencing. So no wonder our understanding of the relationship between microbes and all of our lives and health and disease was so fragmentary, because we basically ignored 95-98% of what is really out there. And when I say out there, I mean really everywhere. There are tons of microbes in soil. There are tons of microbes in our respiratory system. There are tons of microbes, of course, in our gut and in stools. And we are really, again, believe it or not, more microbial than we are human, in terms of at least counting. I'm not trying to make metaphysical assessments here, but the fact of the matter is that every single human being contains approximately between 10, 100, or 1,000, depending on how you count, more microbial cells, bacteria, than human cells. And that's true for all species on this planet. So, we are vastly outnumbered in very many ways by our microbes. And these microbes are enormously diverse. And the diversity is at levels that, again, only now that we can sequence these guys, we begin appreciate. And it's not just the diversity that you can appreciate by sequencing them, is also the biochemical and chemical diversity. So when you sequence and then you do mass spec on the products of these things, you understand what's their functional potential. If you look at their DNA, you understand what their actual capacity at the time in which you sample them from the environment. In other words, with genes they are expressing. So basically what they're making. And then there is this whole blooming, and rightly so, field of metabolomics, because keep this in mind. The main point is this: we have certain areas that are very highly colonized more than others. So skin, and gut, and lungs. The gut by far exceeding probably every other site in terms of how many microbes colonize. But these guys who sit in the gut don't need to travel themselves to influence the function of all other organs in our bodies. All they need to do is make their metabolites. And these metabolites are very small molecules that typically travel probably in ways that we are only now starting to understand, and that probably have receptors that we only now are starting to decipher. That we as humans, and not just we as mammals, have a very large class of cellular receptors, the G protein couple receptors, that have been orphaned for decades. In other words, the ligands for which weren't known. We didn't know what they bound, we didn't know what they did. And now we start finding, oh, by the way, that receptor on that cell that we thought was an orphan receptor is actually the receptor for this metabolite produced by this class of bacteria. Oh, by the way, the other receptor on this other cell type is also a receptor for metabolites. So in essence, what I'm telling you is that all our microbes have to do, in a sense, is sit in the in the gut and produce all these metabolites, and those travel. And those create these axes that are now very much understudied, the gut-lung axis, the gut-brain axis, and so on and so forth. And so in essence, you won't be surprised now to hear that there is virtually no, almost no trait, phenotype, call it what you want, and disease in our body that is not under some level of influence from microbes. So our pressure is largely determined by microbes, because you know what? The way we metabolize salt, we typically stay away from salt if we have high pressure. Well, it's not the salt per se, it's the way that salt is metabolized by salt metabolizing bacteria in the gut. Our weight is enormously dependent on microbes, and I will get back to that. I will show you some experiments that show that conclusively. Microbes change with age. I told you that it's critical what we have as born children. Our microbiome, which is the assembly of all the microbes in our body, changes as we age. Even jet lag is apparently a function of changes in our microbiome induced by a different chronological or chronobiologic clock that we expose ourselves to as we travel. Not to mention mood. Mood is very, very strongly influenced by microbial metabolites. And when we go to diseases, well, that's my list, but you know, it's striking that there are individuals with cancer who respond very positively, and I'm taking an extreme case to make my point, who respond very positively to, for instance, what this class of checkpoint inhibitors that are becoming more and more popular, and rightly so, because they can have incredibly good results in some people. And then there are people that do not respond to them at all. And it looks like, based on the type of studies that I'm going to outline for you, the reason is in the microbiome of these individuals, or at least to a large extent. So, back to our story. My interest is always in causality and never in associations. And so the way to ask to what extent the microbes in, say, an Amish child are responsible for protecting that child from asthma is to really try and do this mechanistically in a way that shows causes. And for that, again, we go back to mice, and that's admittedly a limitation. But again, we can do things in mice we can do in humans. And in particular, we can use mice that are born and kept germ-free. In other words, these are mice that don't have a single microbe, and because they are grown, raised, and kept in these places. These places look like a Martian home to me. None of us could get into those places unless you are heavily trained. You see all those containers and things. That's where you keep your germ-free mice. And these are all containment measures to make sure that not a single bug gets into these mice. Because what you want to do with these mice is that you want to colonize them with the kind of microbes that you want to study. And you can humanize them. In other words, you can take germ-free mice, give them gut microbes from a human being, and they will develop in very, very, very large proportion, not 100%, but very, very close to it, actually, if you do it well. And you get your humanized mouse that has now the microbiome of a Hutterite child or of an Amish child. This became first famous, rightly so, and attracted attention because of this experiment that I want to show you briefly, because it's a pretty extraordinary, Jeff Gordon's group, that showed that you can transfer obesity. I taught you about weight being so important, microbes being so important for weight. You can transfer obesity, transferring microbiota. They took stools from groups of twins, one of whom, so twins that are, however, discordant for obesity. These are identical twins, genetically identical twins, discordant for obesity. And here is what they did. They took the obese microbiome and the lean microbiome. They gave them to germ-free mice, and lo and behold, what they got was a very slim mouse when colonized with the microbiota, stool microbiota from the slim twin, and the very obese one when the mouse had been colonized, germ-free had been colonized with the microbiota from the obese twin. So this is, I'm getting basically to the end, and this is what we think is going on. And this is how we think about the farm effect in my group and in a group of like-minded people. We think that it's all a matter of composition and timing for both immune responses and development of microbiota, especially in the gut, but not only in the gut. And we think it's an interplay between endogenous microbes and environmental microbes. Each of us starts with very few, if any, microbes at birth that, however, are at the time of birth and possibly before birth, transmitted by the mother during pregnancy. And these microbes get to the gut, which is essentially an empty or semi-empty pristine niche, looking forward to being colonized. So during the first year, there is a rambunctious explosion and development of these microbes that accumulate very rapidly and very efficiently in the first year. But they don't do that in isolation. They do it with the constant feedback and input coming from the environmental microbes. And of course, based on what we think is going on and what I just showed you, depending on whether you are in a protected environment or not, the feedback that you get from these microbes will be protective or not. And the same is true, by the way, in terms of what the mother herself transmits to you. And you know, the difference that this makes is this, and it might look evanescent to you, but is not. It's a matter really of timing and composition. The microbiome and immune maturation go hand in hand, and they go from utero to the baby, to the child, and then an adult, and then as you age, goes down. And what I want you to notice is this. This is the sort of healthy curve that you would see in an Amish child, or in a child not Amish who's healthy. This is the allergic asthmatic Hutterite curve. And the point that is that is reached is shifted. I don't know how to do this graphically, but I think you get my meaning. It's shifted to the right. This curve is delayed. This system matures more slowly in these children and to a lesser extent. And it is this delayed and decreased level of immune and microbiome maturation that creates the immunological problems that allow these diseases to emerge. And depending on genetic makeup, depending on environment, and depending on other factors, the diet and everything else, will promote or prevent the emergence of disease. This is how we think about this now: composition, timing, and for both immunity and our microbiological components. So I think this article in the New York Times a few years back, and the statement that some of my best friends are germs, was prescient. I would subscribe to this entirely, but even more, I would subscribe to what my good friend at Caltech, Sarkis Masmanian, has written, that we are fortunately not alone. We wouldn't be who we are and what we are if we didn't have our microbes to work on our behalf. And with this, I'm going to stop, and I'm thanking you for your attention, and please, I will try to ask any question you might have as best I can. Thank you.
Donata, thank you so much. That was fascinating, very well done presentation, very interesting. And I see there are a couple of questions in the Q&A area. I don't know if you can get to those.
Let me see.
There's one from Jack Hughes. One.
Let me stop. Do you need to, can I stop sharing for a moment? I can always go back.
Sure.
Okay, because I can see the chat this way, Q&A. Okay.
Yeah.
John, this is a standing presentation. Thank you. The complex environment of multiple organs in regard to the antigen bacteria becomes even more complicated as we think of relationship of viruses to bacteria that modify their behavior. Would you like to comment further on this degree of complexity? I can comment in a not particularly informative way, because our understanding of the interaction between viruses and bacteria as it pertains to human phenotypes is by no means as advanced as our understanding of them in isolation, but certainly we know that such an interaction is there. I think we don't get me wrong, but I think we need to be a little patient here, because again, our ability to really deconvolute the complexity of the microbial environment is only very recent. And it's actually even increasing, because as you can imagine, at the beginning, the only thing we could do was the very basic, what we call 16S RNA sequencing, which basically tells you who is there and when, but doesn't tell you who is doing what to whom and why. Now we can, at the same time, do a much more extensive analysis of RNA, transcriptomics, and all that, but all of this is taking time. So we are, in a sense, in the infancy of these studies, but certainly, the fact that the microbial world interacts not just with us as hosts, but also with the viral one, that's a critical point and will have to be studied. There is a question, are you doing any research on sarcoidosis of the lungs? Unfortunately not. So I wouldn't be able to give you any intelligent answer. So I'd rather stall on this one. I don't want to say anything wrong. Richard is asking, is microbiome related to autoimmune diseases? Indeed. That's an enormously rich area of research right now. Rheumatoid arthritis, lupus. Anytime, okay, this might be a biased answer, but anytime there is an immune response involved, you need to think about microbes. And it so happens that, of course, and this is not because we immunologists are imperialists in our views, I think, but I think that there is now a full appreciation of the fact that inflammation is an immunologic process. And so to the extent that there is inflammation in a disease or in any trait, you would think that that has something to do ultimately with microbes. So, Leslie, I don't see your question, Leslie.
Down below, I think.
Yes. Do you think it will be possible someday soon to inoculate infants or older children with appropriate microbiome to protect them from asthma? We certainly think so. And that's why we are working towards trying to, we and others, trying to develop these environmental, identify these environmental components that are protective. Now, one thing to keep in mind is this. Going back to this research still is in infancy theme. What most people are advocating today, sometimes very vocally, are these treatments where you take one bug and you give that single bug to someone for some reason. I personally considered that very interesting, but not probably very final and certainly very effective in the long term. Because one thing that we should never forget is that microbes live in communities. And there are reasons why they do that, because there are microbes whose only but very fundamental task seems to be not that much to do things for themselves or for the host, but for the other microbes. And so, in other words, they make it possible for other microbes to have all the nutrients and all the conditions that are necessary. So this is pretty much an ecological perspective that we need to incorporate in our thinking. And it's only after we do that, I think, that we will be able to design experiments. But ultimately, yes, you would want to make sure that you can, if you have reasons to believe that there has been a deprogramming or an altered programming of immune responsiveness in a child, you would want to be able to try and put that response on the right track from the very, very get-go. And finally, so the probiotics that people take are not really helpful. Well, you know, probably I wouldn't say that they hurt, but certainly they are not the whole story, and that's for sure. They are based on, let's put it this way, they are the low-hanging fruits. We know that there are some lactobacilli that are good, and so, you know, you take lactobacilli. But there are situations in which there are certain kinds of lactobacilli, certainly not the ones that are in probiotics, but that seem to be actually associated with harmful effects. So there are lactobacilli and lactobacilli, and there are staphylococci and staphylococci. And this is true. It's all contextual pretty much. And so we need to know more before we can appreciate the depth of this contextuality, but I think we should never forget that it is there. Belinda. Wait a second, one more, if I may. Belinda, I cannot not answer Belinda, who has been working hard to get me to this point. Is work being done on trying to transfer microbes from person who responds well to cancer drugs to one who is struggling to respond? It has not been done as that I know in humans, but these experiments have been done in germ-free mice, and the results are crystal clear. There were 3 papers published the same week, 2 I think in Science and 1 in Cell, showing all the same thing, that you can transfer responsiveness to checkpoint inhibitors by transferring microbiota, and you could transfer non-responsiveness by transferring human microbiota, which is a good thing, because, you know, I mean, if you can turn someone who would greatly benefit from a given treatment into someone and cannot respond to someone who actually does respond, that would probably be a very desirable outcome. Of course, we have to learn how to do this safely, reproducibly, robustly. There are a lot of practical issues to deal with, but I think it's a fantastic adventure that we are all trying to undertake.
Thank you so much. That's fascinating, and it was a wonderful talk.
Thank you.
Dr. Vercelli, we have one in the chat who is asking a question. This is from Diane, and
yes,
you can see that question.
At no time did mothers ever request a special diet for their children's allergies, but in my 3rd generation, my G children, friends in out of school, appear to have so many. Yes. That's a very good question. This is something that puzzles me greatly. It is certainly the case that part of the reason we see many more allergies, and including allergies to food, is that on the one hand, there is a general rise in allergies because of the reasons that, because we have lost our microbes, we have changed our relationship with the environment, we have changed our closeness with the environment, and in part because probably foods are much more processed than they used to be. I mean, now you eat something and you don't really know what you're eating, because you don't know how that, whatever it is that you're eating, was raised or grown. So that's a possibility. How much of that is perception and how much of that is real, it's hard to know, but certainly I think that in the end, you know, we have to study these things and separate wheat from chaff, and try to understand what is there or not. But certainly all of these differences in dietary requirements are something that might, in general terms, go with this increase in asthma and allergies that we see in more recent years. So to some extent, it's justified.
Well,
yes,
Dr. Vercelli, thank you very, very much. This has been fascinating, and we really appreciate your sharing of the time.
Yes.
And we will be in touch with you later regarding whether we or not we post the video. So thank you once again.
I am very grateful for the invitation. It was a lot of fun. Thank you for picking me, and be
Excellent.
well and safe, all of you. Thank you.
Excellent presentation.
Thank you.
Yes.
Thank you.
Yes.
You be well.
Donata, wonderful. Thank you.
Yes.
Thank you. Thank you. Thank you. Thank you all.
Yes.
Thank you.
Yes. Very
Bye-bye.
good.
Your day.
Bye.